Going to understand structure and evolutionary significance of Echinoderm larvae. So it will be an introduction part for the Echinoderm larvae. Echinoderms, as we know, they are aquatic organisms purely marine. They live in seawater, so all the developmental process occurs within the water. We have seen that the Echinoderm adults are said to have no sexual dimorphism. They both, the male and female, look similar. Second important point is that both male and female, even though they are separate, even though they don't have the sexual dimorphism, they are separate. So this condition we call it as unisexual or dioecious or gonochoristic condition, which we have learned in the previous class. The male organism produces at the time of breeding the sperms as the gametic cells, which are said to be dinoflagellate in condition. Females also produce their gametic cells called the ova, which is said to have the covering of vitellite membrane, and both said to have the pronucleus of male and female respectively in their gametic cells. After the production of their gametes, they are released into the water, and in the water there occurs the phenomenon of external fertilization. Mean to say, the male gametes and female gametes undergo a fusion process, which we call it as external fertilization. So after the fertilization process, the cell is called zygotic cell, which is diploid in nature. This diploid cell called zygote now will undergo a mitotic division, which we call it as cleavage. Cleavage is a phenomenon where the mitotic divisions are said to be holoblastic, mean to say it completely divides the cells. They are radial, the plane of division. They are equal, the cell size after the division will remain same, and they are indeterminate because after the formation of the division of cells, even if we remove the cells from the mass, they are capable of forming the complete organism. So hence it is called indeterminate. So this is the type of cleavage that we find within the organism. After we understand the cleavage process, the cleaved cells now forms a mass of cells which are referred as the morula stage. After the formation of morula, the cells now shows the next stage called coeloblastula. Coeloblastula is the specific stage of echinoderms where all the cells will form a single layer of ciliated epithelium having a cavity called blastocele filled with the coelomic fluid. This ciliated cell helps the embryonic stage to have free movement in the water. After coeloblastula stage, the next immediate stage is called gastrula. In gastrula, there occurs invagination at posterior mid-posterior region of Blastoderm undergoes invagination and forms a new cavity which we call it as orchenteron. So after the new cavity is formed, the cell layer inside is called endoderm. The outer layer now is called ectoderm. From the endoderm layer, the cells will separate out, which we call it as delamination, and they form a new layer in the center between the ectoderm and endoderm, which we call it as mesoderm. So three germ layers are formed, namely outer ectoderm, middle mesoderm and inner endoderm for this particular stage of development. Students, remember this particular cavity, what the invagination of endoderm encloses is called orchenteron, and the opening of this orchenteron towards the outer environment is called as the blastopore, which is responsible for the formation of anus in the developmental stages. So after gastrula, then comes the indirect developmental aspect of the echinoderm larvae. All echinoderm larvae will show a common indirect developmental larval stage called dipurula. Since we have echinoderm showing external fertilization, the development is indirect. It includes the larval stages. The common larva of most of the echinoderms is dipurula larva. This dipurula larva will have a ovoidal shaped body which is transparent, having a ciliated band towards anterior nearer to the mouth called preoral ciliary band. The one which is nearer to the anus is called as postoral ciliary band. And the ovoidal shaped dipleural larva will have anterior-most zone called as preoral loop and posterior is called as postoral loop. It will have a complete digestive system where it will have mouth towards one end and anus towards the other end. So it will have the complete digestive system also. The larvae are generally said to be plantotrophic, mean to say they will feed only on the planktons, the microscopic plants and animals present inside the water. So this larva is a pelagic larva. It floats freely in the sea water. It is transparent, free swimming, pelagic larva, and it is said to have the complete digestive system which is responsible to have the process of plantotrophic nature for the dipleurula. Now this dipleurula after some time undergoes the clockwise rotation and forms the different larvae of different class of echinoderms. In case of Asteroidea, it forms Bipinnaria larva. In case of Holothuroidea, it forms Auricularia larva. In case of Echinoidea, it forms Echinopluteus larva. In case of Ophiuroidea, it forms Ophiopluteus larva. In case of Crinoidea, it forms Dolalia larva and Pentacrinate larva. So when we look at the larvae, almost all the larvae of every class of echinoderms will have all these features of development in common. And this particular larvae is whatever we have, and they have the common features which is said to be bilaterally symmetrical. So all larvae are said to have bilateral symmetry, and none of the adult characters of echinoderms are noticed in the development of either larvae or the developmental early developmental stage of the larvae. Thereby, the larvae of echinoderms are generally called as primary larvae. So remember, primary larvae are those larvae which doesn't show any of the adult characters in their larval forms. So primary larvae are found in the larvae of echinoderms. So this is about the general introduction related to formation of echinoderm larvae.